In this video, let us look at the cells of the epidermis. So, generally, the skin is covered by what you call stratified squamous keratinized epithelium. This offers protection to the skin. Alright, so the cells which you find within the epidermis, they include keratinocytes, melanocytes, Langerhans cells, and Merkel cells. So the keratinocytes are most abundant within the skin. They are the cells which make up the layers of the epidermis. Alright, so they're the cells which make up the stratum basal, the cells which make up the stratum spinosum, stratum granulosum, lucidum, and corneum. And then within these keratinocytes, or interspersed between these keratinocytes, that's where we may find the other cells like melanocytes, Langerhans cells, and Merkel cells. Okay, so let's start with keratinocytes. Keratinocytes, they originate in the stratum basal, where we have the stem cells or the progenitor cells for the keratinocytes. Okay, and these cells, they undergo differentiation until they reach the stratum corneum and then become fully keratinized and dead. All right, so let's look at this differentiation. In the stratum basal, they synthesize keratins. All right, and so for the reason that they synthesize keratins, they have more ribosomes. All right, and then these ribosomes give the cells of the stratum basal a basophilic staining. Right. Within the stratum spinosum, the synthesized filaments, they will sort of like bundle together to form what you call tonofibrils. Right. So these tonofibrils, they will accumulate in the cytoplasm and give the cytoplasm a characteristic eosinophilic staining. All right. Now, when they enter the stratum granulosum, these cells, they will synthesize granules, and these granules are called keratohyalin granules. They contain two main components, namely filaggrin and trichohyalin. So these keratohyalin granules, they facilitate aggregation of keratin filaments within the cytoplasms of these cells. Alright, so they will facilitate the process which you call keratinization of the cells. Alright, cells in the stratum granulosum, they form what you call an epidermal water barrier. Alright, this one is our epidermal water barrier here. So the epidermal water barrier is formed from lamellar bodies which are synthesized within the cytoplasm of keratinocytes in the stratum granulosum, and an exterior lipid membrane which will attach to the cell membrane from outside, right? So the epidermal water barrier acts as a permeability barrier. It prevents the excessive loss or uptake of water by the skin, but at the same time it allows lipid-soluble substances to pass through it. Okay. Now, for the cells within the stratum lucidum, these cells, they are anucleate, they do not have cell organelles, they are dead, and they are keratinized. So we see that at the uppermost layers of the stratum granulosum, the cells begin to undergo terminal differentiation, where they lose their nuclei, their organelles, and then they become dead cells. So these cells, they will accumulate keratin filaments within the last two layers of the skin, which is the stratum lucidum and the stratum corneum. Then these keratin filaments will fill out the cytoplasm and give the cells a characteristic eosinophilic appearance. Alright, so these cells, they are squamated. As you can see, these cells, they are squamated cells. Okay, and at the uppermost layers of the stratum corneum, these cells begin undergoing a process known as desquamation, where they are removed from the surface of the skin. Right. So recall that the skin is continuously reforming. Right. So in order to maintain a constant thickness, there has to be a balance between the addition of new cells and the removal of old cells. So at the uppermost layers, the cells, they are sloughed off from the surface or removed from the surface. All right. So the skin thickness is maintained. Okay. Now let us look at melanocytes. These are cells within the epidermis, specifically within the basal layer of the epidermis, which is the stratum basale, which synthesize melanin. Alright. So these cells, they are neurocrest derivatives, and they appear histologically as clear cells, okay, with rounded cell bodies and the central nuclei. Okay, they are attached to the basal lamina by the hemidesmosomes, but however, they lack individual attachments to their adjacent keratinocytes. So these cells, they have long cytoplasmic extensions, all right, which will extend between the keratinocytes in the layers in the stratum basale, in the stratum spinosum, such that when these melanocytes synthesize their melanosomes, they will travel within the cytoplasmic extensions to various cells in various layers of the epidermis. Right? So each melanocyte and the keratinocytes to which it supplies melanin is actually referred to as the epidermal melanin unit. Alright, so for the identification of melanocytes, we can find them in the stratum basale, and they are clear cells. So they are different from other cells of the stratum basale, because the other cells, they appear basophilic. These ones, the keratinocytes, they appear basophilic, but these ones, they are clear cells. They have a clear cytoplasm. All right. The Langerhans cell, these are antigen-presenting cells derived from monocytes, and they are much more abundant and clearly seen within the stratum spinosum. These ones, they have cytoplasmic processes which extend between the cells of the layers of the stratum spinosum, and these antigen presenting cells, they catch the antigen, they modify it, and then they present it to the lymphocytes, which will then elicit an immune response, right, thereby preventing the entry of antigens into the deeper layers of the skin or into the body. So we can identify Langerhans cells by their clear cytoplasms within or in between the cells of the stratum spinosum. These ones, Langerhans cells, right, in the stratum spinosum. All right. The Merkel cells, these are sensory cells located within the stratum basale, right. They are tactile cells, meaning that they sense touch, gentle touch, all right, and they are much more abundant in the highly sensitive skin. So these cells, they have these little processes here which will act to pick up any stimuli and then convert the stimuli to an electric signal, which is picked up by the expanded terminal of an afferent fiber. So for the clinical correlates, we have drug administration via skin patches, and then this bases on the fact that we have that epidermal water barrier. Right. So the epidermal water barrier is selectively permeable. It does not allow water to pass through, but it allows lipid-soluble molecules to pass through. So lipid-soluble drugs such as estrogen can be administered to the body via the skin patch method. Alright, for skin color, we have few factors determining skin color, the first of which is melanin. Alright, so the amount and type of melanin produced by melanocytes determines skin color. It's not the number of melanocytes, but it's the amount of melanin produced by the available melanocytes. So some people produce too much melanin, some people produce too few melanin. Alright, that's the difference. We also have blood. So the blood flow to the skin also adds an element of skin of color to the skin. Then we have UV radiation. So exposure to UV radiation from the sun stimulates melanin production, leading to a darker skin. So those melanocytes, they respond to UV radiation. If UV radiation increases, the rate of production of melanin also increases. Alright, that's why in very hot environments you find people who have much darker skin. Albinism is mainly caused by partial or complete absence of melanin production by the melanocytes. So this is mainly due to the defect in the enzyme tyrosinase, which is required in melanin synthesis. So this person has little to none melanin in their skin, so that is why their skin appears pale. Alright, vitiligo is more of an acquired condition which is characterized by skin depigmentation. Alright, so this depigmentation, it occurs in affected patches of the skin. So not the entire skin is affected, but like certain areas are affected due to either loss or decreased activity of melanocytes. All right.